Considerable evidence that's been accumulated over many years shows that phytonutrients derived from members of the brassica family of the cruciferous vegetables have potent breast cancer chemopreventative properties. Well-known cruciferous vegetables include broccoli, cabbage, cauliflower, and Brussels sprouts. A meta-analysis of data from 11 case control and 2 cohort studies have found that women with higher amounts of cruciferous vegetables in their diet were less likely to be diagnosed with breast cancer. On the basis of this and other evidence, researchers encourage the inclusion of cruciferous vegetables in the diet of women who are diagnosed or at risk of breast cancer. Cruciferous vegetables provide, either directly or through precursors, beneficial compounds. Among these most well studied are sulforaphane, indole 3-carbinol, and di-indolemethane. Sulforaphane is a potent inhibitor of breast cancer cells in lab studies. In vitro research suggests the sulforaphane's anti-breast cancer activity might be in part attributable to the reduced cell proliferation, increased apoptosis, upregulation of detoxifying enzymes in healthy cells, and the downregulation of these detoxifying pathways in the cancer cells, promotion of the cell cycle arrest in cancer cells, and elimination of cancer stem cells, among others. Cell culture studies also showed that sulforaphane increased the expression of the genes that encode 2 proteins which slow tumor growth, phosphatase and tensin homolog, and retinoic acid receptor beta 2. Additionally, the sulforaphane was shown to enhance the effectiveness of the chemotherapy drug 5-fluorouracil and paclitaxel against breast cancer in vitro. Several in vivo animal models of breast cancer have also shown sulforaphane, typically as subcutaneous or intraperitoneal injections, can reduce a tumor mass. Preliminary human clinical evidence suggests the sulforaphane can modulate histone deacetylase activity in the peripheral blood cells from women with abnormal mammograms. This activity influences the acetylation of several of the genes involved in cancer development and progression. Large clinical trials with longer duration of supplementation are needed to clarify the breast cancer chemopreventive effects of the sulforaphane. As of late 2021, one trial ongoing in Texas is testing if the sulforaphane supplementation can reduce the cardiac toxicity of the chemotherapy drug doxorubicin in women who are undergoing breast cancer chemotherapy. Upon ingestion, the cruciferous vegetable constituents called the glucoisonolates are transformed into the isothiocyanates. These are a class of compounds with sulforaphane being among the most thoroughly studied. The plant enzyme myrosinase is responsible for this conversion, and it takes place in the digestive tract. In whole, mature cruciferous vegetables, the sulforaphane exists predominantly in its precursor form, glucoraphanin. When cruciferous vegetables are broken down in the digestive tract, the myrosinase is liberated, and it can interact with the glucoraphanin, and it yields sulforaphane. However, cooking the cruciferous vegetables degrades the myrosinase, and it leads to lower sulforaphane bioavailability from the cooked rather than the raw. Raw broccoli and broccoli sprouts are good sources of glucoraphanin, and the human gut microbiota can also convert the glucoraphanin to sulforaphane, but it's at low efficacy. A significant obstacle in developing clinically effective sulforaphane preparations has been devising a method to provide the more stable glucoraphanin, along with the myrosinase enzyme. Recently, researchers discovered that enterically coated preparations containing both the glucoraphanin and the myrosinase appear to increase the sulforaphane's bioavailability, possibly by protecting the myrosinase from degradation in the acidic environment in the stomach. In lab studies, indole 3-carbinol slowed the proliferation of tumor cells. Moreover, the indole 3-carbinol can induce favorable changes in the levels of different forms of estrogen, promoting the degradation of the estrogen receptor alpha and reducing the activity of an enzyme that synthesizes estrogen. In lab and animal models of hormone-dependent cancers, inducing breast cancer, the indole-3-carbinol showed protection against the tumour proliferation. In a small study that enrolled 5 obese women, supplementation with 400 mg of indole-3-carbinol daily for 2 months led to an increase in the ratio of 2-hydroxyesterone to estradiol. The researchers concluded this response to the indole-3-carbinol might result in a hormonal milieu that helped to reduce the estrogen-dependent cancer risk. Diindolylmethane is a product from the metabolism of indole-3-carbinol. As with the indole-3-carbinol, there's significant lab data suggesting the diindolylmethane has cancer-fighting properties. In mice, the diindolylmethane inhibited the breast cancer formation and it reduced the growth of existing breast cancers. Preclinical research also showed that diindolylmethane increased the radiosensitivity of human breast cancer cells resistant to several drugs. Evidence from human studies also suggests the cruciferous vegetable intake and the indole-3-carbinol diindolylmethane supplementation can be beneficial in the context of breast cancer chemoprevention. One study assessed the dietary intake of cruciferous vegetables in 54 women with abnormal mammogram findings. Among the women, those with the highest levels of the cruciferous vegetables in their diet had the lowest levels of a proliferation marker in their biopsy tissue samples. In a randomized trial of women taking tamoxifen, supplementation with diindolylmethane at 300 mg per day improved several markers of estrogen metabolism. A study of postmenopausal women with early-stage breast cancer had similar beneficial results. Interestingly, the 300 mg per day increased the expression of the BRCA1 mRNA among the breast cancer patients with BRCA1 mutations. It increased the expression of this gene and the subsequent normalization of protein levels, and this might potentially help diminish the effects of the harmful mutation. A small single-arm clinical study evaluated the effects of the diindolylmethane supplementation on breast density in 23 healthy women who carried the BRCA gene mutation. The participants took 100 mg of diindolylmethane every day for a year. At the end of the study, the investigators reported the women who were taking the diindolylmethane exhibited a decrease in the average amount of fibroglandular tissue, which is a measure of breast density. A matched comparison group of women who didn't take the diindolylmethane didn't show a change in the breast density during a parallel year. Silmarin is an extract of milk thistle, and it mainly consists of the lignin silbin. In lab studies, the silmarin blocked proliferation of tumour cells, reduced inflammation, and protected cells from carcinogenesis. Silbin might also improve the effects of chemotherapy. The chemotherapies paclitaxel and doxorubicin destroy breast cancer cells more effectively in the lab when the cells are co-treated with the silbin. One study in humans confirmed that a preparation of the silbin effectively reduced breast cancer tissue, but the effect of the silbin on the breast cancer outcome has not been tested. Curcumin is a bright yellow component of the spice turmeric. In lab studies, curcumin can slow the proliferation of breast cancer cells, cause tumour cells to die, and prevent tumours from developing blood vessels to supply nutrients. Curcumin might also specifically target cancer stem cells, rare tumour cells that form new tumours. Lab studies have also found curcumin can make the breast cancer cells more sensitive to chemotherapy drugs, including 5-fluorouracil, paclitaxel, and doxorubicin. A phase 1 clinical study used a combination of curcumin and docetaxel for patients who had advanced and metastatic breast cancer, and it found the combination was well tolerated. Scientists are looking into the anti-cancer effects of the curcumin, and they found the curcumin alters many signaling pathways that the cancer cells heavily rely on. Inflammation can promote tumour growth. In lab studies, the curcumin can reduce the levels of inflammatory cytokines in breast cancer cells. One pro-inflammatory cytokine is tumour necrosis factor alpha. In lab experiments, the tumour necrosis factor alpha changed the metabolism of the tumour cells in a way that provided them with the energy to sustain the rapid growth. Treating the cancer cells with curcumin blocked the effects of the tumour necrosis factor alpha. Curcumin might also help to fight the way that tumours protect themselves from the immune system. Natural killer cells are often impaired in patients with cancer. Mouse breast cancer cells treated with curcumin cannot suppress the natural killer cells as effectively as the untreated cells. Tumours can also protect themselves using a type of suppressor T cell called a regulatory T cell, or a Treg. The Tregs dampen the immune response. When mice with breast cancer were treated with curcumin, the suppressive capacity of the Tregs was reduced by other T cells, and they were able to destroy the tumour cells. Apigenin is a flavone and is present in fruits and vegetables like onions, orange, tea, celery, artichoke and parsley. It's been shown to possess anti-inflammatory, antioxidant and anti-cancer properties. Many studies have confirmed the cancer chemopreventative effects of the apigenin. Apigenin stimulates the self-destruction or the apoptosis in the breast cancer cells. One study showed the apigenin slowed the progression of human breast cancers in mice by inducing the cell death, inhibiting cell proliferation, and reducing levels of HER2. Blood vessels responsible for feeding the cancer cells were smaller in apigenin-treated mice compared to the untreated mice. Smaller vessels reduced the nutrient flow to the tumours, and they might deprive the cancer of its nutrients. The apigenin can also enhance the effects of chemotherapy drugs. Blueberries are rich in cancer-fighting compounds. Mice with breast cancer who were fed a diet supplemented with blueberry extract powder had smaller tumours and fewer metastases than the mice who were fed a controlled diet. Many lab studies have tested the effects of one particular compound in blueberries, pterostilbene, and it's been found to increase the self-destruction or the apoptosis of the breast cancer cells, reduce the proliferation of 3 subtypes of breast cancer, inhibit metastases, and enhance the effects of tamoxifen. CoQ10 helps cellular enzymes turn food into energy. For several decades, it's been known that cancer patients often have decreased blood levels of CoQ10. These findings sparked interest in the compound as a potential anti-cancer agent. In a clinical study, 32 patients were treated with CoQ10, in addition to other antioxidants and fatty acids. 6 of the patients showed particular tumour regression. In one of these cases, the dose of CoQ10 was increased to 390 milligrams, and within 1 month the tumour was no longer palpable. After another month, mammography confirmed the absence of the tumour. A different patient with tumour tissue remaining after surgery took 300 milligrams of CoQ10, and within 3 months there was no residual tumour tissue. The complete regression of breast tumours in some patients, coupled with the further reports of disappearance of breast cancer metastases in several other cases, demonstrated the potential of CoQ10 as an adjuvant therapy for breast cancer. Conjugated linoleic acid, or CLA, is found mostly in dairy and meat products. It's a type of fat with some health-promoting properties. Some case-controlled studies suggest a diet rich in CLA can reduce the risk of breast cancer. For instance, in one study, postmenopausal women with the highest CLA intake were 60% less likely to develop breast cancer compared to the group with the lowest intake. In one clinical trial, 24 women with stage 1, 2 or 3 breast cancer, scheduled for surgery, were treated with 7.5 grams of CLA per day. The authors found that a marker of cellular proliferation was decreased in the tumours after the treatment compared with before the treatment. Pomegranate is rich in healthy polyphenols, and it's been used for centuries for medicinal purposes. Researchers discovered that treatment with the whole pomegranate seed oil and the juice concentrate resulted in dramatic growth inhibition of estrogen-dependent breast cancer cells in the lab. The same study showed the inhibition of tumour formation in rodent cells who were exposed to chemicals known to cause breast cancer. Using different methods, another research group found a 42% reduction in tumour formation in the mice with pomegranate juice polyphenols, and an 87% reduction with pomegranate seed oil. Pomegranate seed oil is a potent inhibitor of aromatase, the enzyme that converts testosterone into estrogen. This enzymatic blockade could contribute to the pomegranate seed oil's ability to inhibit the growth of estrogen-dependent breast cancer cells. The pomegranate extract has also been shown to enhance the effects of estrogen-blocking drug tamoxifen and to reduce levels of estrogen receptors in the tumours. Pomegranate also increases apoptosis, even in cancer cells that lack estrogen receptors. Cancer cells need to grow new blood vessels to support their rapid growth and tissue invasion. They typically do this by ramping up the production of certain growth factors, like vascular endothelial growth factor. Pomegranate seed oil powerfully inhibits the production of this growth factor. In a lab model of vessel growth, these modulations translated into a significant decrease in new blood vessel formation. The pomegranate seed oil contains a number of unique chemical constituents with potent biological effects. Punic acid, an omega-5 polyunsaturated fatty acid, inhibited both estrogen-dependent and estrogen-independent breast cancer cell proliferation in lab cultures, and it induced apoptosis at rates of up to 91% higher than those in the untreated cell cultures. Quercetin is a compound and it belongs to a group of pigments called flavonoids. It's found in a broad range of plant foods, from grape skins and red onions to apples, green tea, and tomatoes. Quercetin can protect the DNA from cancer-inducing mutations. In breast cancer cells, the quercetin stops tumor cells from dividing, and it causes them to self-destruct. These effects inhibit the growth of tumors in mice, and they prolong the survival of mice with breast cancer. Furthermore, the quercetin favorably changes the chemical signaling pathways that are abnormal in cancer cells. For more on breast cancer, herbs, supplements, or natural treatment plans, check out my website.